So I'm going to take a moment here to review the biochemistry of contraction. So essentially, what happens here is that calcium, which I circled here, enters via the L-type calcium channels. Calcium then binds to calmodulin, which then leads to activation of MLCK, which is also known as myosin light chain kinase, which as a kinase means it's an enzyme that essentially phosphorylates. So MLCK phosphorylates myosin. Myosin, when phosphorylated, is able to connect to actin to lead to contraction. Additionally, these cells, these smooth muscle cells, contain an internal storage of calcium called sarcoplasmic-reticulum, which is SR right in the middle. SR also releases calcium, which binds to calcium calmodulin, leading to the same pathway. So you can imagine when there's a decrease in calcium, you have decreased phosphorylation, and therefore decreased contraction. But what about that myosin that's already phosphorylated? Well, you have something called MLCP, which is shown in the bottom right, which stands for myosin light chain phosphorylation inhibitor, essentially. So MLCP is, or not phosphorylated, it's myosin light chain phosphatase. I apologize. MLCP goes to phosphorylated myosin and takes away that phosphorus, and therefore is able to lead to the separation of myosin from actin. So if you look at all these this pathway, how exactly do these receptors, as well as molecules that I mentioned in the previous slide, get put into it? Well, on the bottom right, alpha-1 receptors, vasopressin 1 receptors, endothelin and angiotensin inhibit myosin light chain phosphatase. So by inhibiting that, you have a longer time in which myosin is phosphorylated and therefore increased contraction. On the other hand, nitric oxide, which is produced in the endothelium, activates myosin light chain phosphorylase. Now, on the left side, you can see that obviously calcium channel blockers would inhibit the ability for calcium from entering the cell. Beta 2 agonist right in the middle inhibits myosin light chain kinase and therefore would inhibit contraction. Now, you might say, well, how does alpha 2 fit into all this? So alpha 2 has two effects. Alpha 2 activates MLCK, which you would expect to lead to increased contraction. However, in the presynaptic neurons, you also have alpha 2 receptors. And these alpha 2 receptors inhibit the ability for signals that lead to increased calcium influx into the smooth muscle cell. So therefore what happens is the alpha 2 presynaptic effect, which again inhibits decreases essentially norepinephrine release from the neuron to the smooth muscle cell, overwhelms any kind of effect that the alpha 2 may have directly on MLCK. And as a result, alpha 2 agonists, such as clonidine, actually leads to vasodilatation and therefore hypotension. Okay? Hence my comment on the bottom left that says presynaptic effect is great.
Guys, thank you very much for watching. Make sure that you like, subscribe, and share this video. Like it if you like it. If you don't like it, then don't like it. And then subscribe if you want more videos. We upload videos regularly, so if you subscribe to the channel, you can get a notification and the video will appear in your inbox. And if you hit the bell button as well, then you can get the notifications for this as well.